Hello there. Today we're going to discuss the nervous system. These are the objectives for this unit. First, we'll start with microscopic anatomy and physiology of the nervous system. Neurons is the basic cell of the nervous system. It assumes a variety of shapes and sizes, but even so, there's three basic parts to a neuron. The cell body, also called the soma, shown here, is the control center and contains the nucleus. Actually, the soma or body is here. And then the second part is the dendrites, which are these structures branching out from the cell body. The third part is the axon, shown here, and an important part of the axon is the synaptic knobs or the synaptic terminal down in this region, and that will become more evident in a minute when we discuss that. Dendrites receive signals from other neurons through the dendrites here coming in, and they send information to other neurons through the axon out to the synaptic terminal going that direction. Some neurons have only one dendrite and some have thousands. The axon, most neurons have only one axon. Occasionally they might have a collateral axon here that comes in and hooks into the axon, but nonetheless there's just one axon extending from the cell body. The length of the axon ranges from a few millimeters up to a meter long. The axons of many but not all neurons is encased by a myelin sheath, which are these little structures here with these gap areas in between. The myelin sheath encases and insulates the axon. It's composed mostly of lipid, and it's secreted in the peripheral nervous system by Schwann cells. In the central nervous system, it's created by oligodendrocytes. These little gaps that I talked about in between the myelin are called nodes of Ranvier, or gap junctions, and that's these little things right here. Within the synaptic knob at the end of the axon are little vesicles containing a neurotransmitter. The neurons handle the nervous system's role of communication. There's three basic types: sensory, interneurons, and motor neurons. Sensory neurons detect stimuli such as touch, pressure, heat, cold, or chemicals, and they transmit stimuli, information about stimuli to the central nervous system. Interneurons are found only in the central nervous system, and they connect the incoming pathways with the outgoing motor pathways. So incoming pathways are called sensory, outgoing pathways are called motor. The motor neurons relay messages from the brain to the muscle or glandular cells. Neuroglia, also called glial cells, are the supportive cells of the nervous system. Neurons are high-maintenance cells, kind of like a Ferrari. They do their job really well, but they require a lot of maintenance so that they can perform their function. These cells perform various functions to enhance the performance of the nervous system, and frankly, just so that it can survive. Other than Schwann cells down here, This is the Schwann cell. The other four types are found only in the central nervous system. Schwann cells are found in the peripheral nervous system. Astrocytes here are the most numerous of all of the glial cells and pervasive throughout the brain. These tiny little feet, these little processes are called foot processes that go out and they surround, you can see here, here's another one, it surrounds the capillaries in the brain. Others connect, other of these processes connect to other neurons, like this one here will go out and connect with another neuron. These projections that wrap around capillaries form the blood-brain barrier, and they funnel glucose and other nutrients from the bloodstream to the neuron. They're also important for filtering potentially harmful substances so that they don't ever get into the neurons and destroy them. The blood-brain barrier is a semi-permeable membrane, and it's permeable to small molecules, again, like oxygen, carbon dioxide, and water, but it blocks larger molecules like proteins and bacteria and other pathogens. The blood-brain barrier also prevents most medications from reaching brain tissue, which is a very important function of the astrocytes. In the peripheral nervous system, we have Schwann cells, which are shown here in this figure. These are Schwann cells. These over here are oligodendrocytes. Schwann cells actually wrap themselves around the axon, kind of like a cinnamon roll. They lay down multiple layers, shown here, starting in the middle and coming out towards the periphery. The inside layers form the myelin sheath. The nucleus and most of the cytoplasm of the Schwann cells are located in the outermost layer out here. So this is full of cytoplasm and the cell nucleus, which is incorporated into the actual myelin sheath. This outer layer that I just spoke of is called the neurolemma, and it is essential for injured nerves to regenerate themselves, this neurolemma. If it's missing, that cannot happen. In the central nervous system, projections from one oligodendrocyte, shown here, these projections come out to several nerves and wrap themselves around the nerves, specifically the axons of the nerves. Because the nucleus is located away from the myelin sheath, there's no neurolemma as there is in Schwann cells. And this is what prevents injured neurons in the central nervous system from regenerating. Nerves in the peripheral nervous system are able to regenerate because of the neurolemma. The myelin is very important. As I mentioned earlier, it's a lipid substance that insulates and encases the nerve, sort of like copper wire has a plastic sheath around it. It's the same function. Unmyelinated nerves, we find those also in the nervous system, but typically they perform functions where speed is not so essential. These nerves are usually part of the autonomic nervous system and do things like stimulate the secretion of stomach acid. But the nerve fibers that run to the skeletal muscles, speed is critical for those. Myelination begins during the 14th week of fetal development, but it's not complete until late adolescence, usually around the age of 17 or 18 years old. Impulse transmission results from the flow of electrical charge particles from one point to another across the cell membrane. Be sure to watch this video that you can access through the lecture slides because it provides a really good overview. In the body, whenever ions with opposite electrical charges are separated by a membrane, an electrical potential exists, and it's called a membrane potential. A membrane that exhibits a membrane potential has an excess of positive ions on one side and an excess of negative ions on the other side, and it's said to be polarized. In reality, it's not so much negative ions on the inside, but there is more positive ions on the outside relative to the inside, which creates a net positive charge out here on the outside of the membrane and a net negative charge on the inside. And this potential is established by the sodium-potassium pump that we talked about, which is constantly running using ATP to establish this electrical gradient. When a neuron is not conducting an electrical signal, the interior has potassium, but there's much fewer potassium ions on the inside relative to the sodium ions on the outside. Because of the membrane permeability, a certain amount of sodium leaks back into the cell and a certain amount of potassium leaks out of the cell. So this membrane pump, the sodium-potassium pump, is critical for maintaining this electrical gradient. So what happens is a stimulus, such as chemicals, heat, or mechanical pressure, causes channels on the neuron's membrane at the axon hillock, and these are called localized potentials. So what these do, they're shown here in this diagram or graph, is these stimulus-gated sodium channels. If there's enough of these localized potentials to go from the resting potential at minus 70 millivolts, if it's enough to reach threshold and barely cross threshold, then an action potential occurs, and this is an all or nothing response. And these are voltage-gated sodium channels. So these are sensitive to voltage. Again, in this area, it's stimulus-gated. So any stimulus will open the sodium channels, anything like, again, heat, chemicals, or mechanical pressure. In this area, it's voltage-gated sodium channels that open. And during this phase, it's depolarization. Sodium ions are rushing into the cell down their concentration gradient. This creates a positive voltage change. So it goes from about minus 57 here is threshold, all the way to plus 30 millivolts. This is the measuring the voltage on the inside of the cell in here relative to the outside. You can see that the one electrode is on the inside and one is on the outside. Once it reaches 30 millivolts, the voltage-gated sodium channels close and the potassium voltage-gated channels open here. The potassium then rushes out of the cell down its concentration gradient, and this is the phase called repolarization here. It goes all the way down to minus 70 and even exceeds it a little bit, and this is called repolarization. The reason that the voltage drops below minus 70 millivolts is that sometimes the potassium channels stay open a little bit longer and more potassium rushes out of the cell, And so during this phase right here, until the sodium-potassium pumps can reestablish the resting potential in this region, you have a slight overshoot. And then it goes back to resting membrane potential until another stimulus or group of stimuli open the stimulus-gated, sensitive sodium channels and allows them to go above threshold. This will all make sense more on the next slide when I show you the anatomy of a neuron. So another term for an action potential is a nerve impulse, and an action potential again is this combined region here. This is a nerve impulse, and this is what is this electrical signal that's propagated down the axon of the neuron. One other important point is that at the very end of the action potential, after repolarization has occurred, during this region that we talked about right here, while the sodium-potassium pump is reestablishing the resting potential, this is called a refractory period. And if the nerve receives another stimulus, say in this area, it will not respond until it's been able to reestablish the resting membrane potential here. So there's a refractory period of a little bit less than a second, several milliseconds, where the nerve cannot fire again. And again, that's known as the refractory period. Myelin is very important because it blocks the sodium-potassium pump. It blocks the free movement of ions across the membrane, and so the sodium and potassium do not establish any sort of gradient in the areas where there's myelin here. That only occurs at the gap junctions or nodes of Ranvier. This area right here is the axon hillock, and this is where the stimulus sodium gates are located. Again, that's the axon hillock. This is where localized potentials, localized stimuli affect the neuron, and once there's enough to cross threshold, then the action potential is propagated down the axon in this direction. So to reiterate, electrical changes occur only at the nodes of Ranvier here. The current the electrical current flows under the myelin here until it reaches a node. Once it reaches a node, the action potential is amplified, and so the electrical current flows under the myelin to the next gap junction, where an action potential is again stimulated. Because the action potentials only occur at the gaps, the impulse appears to leap from one node to another, and this type of conduction is called saltatory conduction, and that's what these arrows here and here are representing. The movement of the impulse down the axon is similar to electricity being transported over long distances from an electrical generating facility such as a dam to a metropolitan area. Along the way, there's transformer stations, and their purpose is to magnify or boost the voltage up to where it needs to be, so that the power is not lost during the transportation process. And this is precisely what the gap junctions do. They boost or reestablish the voltage so that the electrical impulse can continue down the axon. As the impulse reaches the synaptic knobs at the end of the neuron, several things happen. They need to pass a synapse, or pass across a synapse, not through but across here. So this is an actual physical gap between the synaptic knob on one end and a dendrite of a neighboring neuron. So when an action potential reaches the synaptic knob, the membrane depolarizes, the ion channels open, but these ion channels allow the passage only of calcium instead of sodium. So you have calcium at the synaptic knob that rushes into the axon. The infusion of calcium causes vesicles, shown here, that are full of neurotransmitter to then move to the membrane, and through exocytosis they release the neurotransmitter into the synaptic cleft. The neurotransmitter then binds to receptors on the postsynaptic membrane, which is part of the dendrite of the receiving neuron. Each neurotransmitter has a specific receptor. For example, the neurotransmitter epinephrine can only bind to receptors specific for epinephrine. The specific neurotransmitter determines whether the impulse continues, called excitation, or whether the impulse is stopped through a process called inhibition. So these neurotransmitters can actually prevent a stimulus from propagating through inhibition. If the impulse is inhibitory, the potassium channels open and the impulse stops. So it would be potassium channels on the dendrite that would open, and potassium would rush out, and that would prevent the impulse from propagating. In excitation, which is shown here, the sodium channels are opened on the dendrite so that the action potential can continue. The receptor then releases the neurotransmitter, after which it's reabsorbed by the synaptic knobs and it's recycled or destroyed by enzymes, shown here. So once it's done its job, it's released, and an enzyme, shown here, breaks apart the neurotransmitter. Sometimes what happens is the neurotransmitter is actually reabsorbed through endocytosis back into the synaptic knob, where it can be reused and recycled. So sometimes it's destroyed here in the synaptic cleft, other times it's reabsorbed. There's an excellent video covering overview of synapses that you can access through the lecture slides. Be sure to watch that. Now we're going to talk about the gross anatomy and physiology of the nervous system, and we're going to start with the central nervous system. The spinal cord is one part of the central nervous system. The other part, of course, is the brain. The spinal cord relays messages from the brain to the rest of the body, and it extends from the base of the brain right here at the bottom of the brainstem until about the first lumbar vertebrae down here. A bundle of nerve roots, small fibers called the cauda equina, which is here, extends from the end of the spinal cord down to the coccyx. The filum terminale is the area where the cauda equina inserts into the coccyx, and this is sort of a tethering of the spinal cord. One important thing to remember is spinal anesthesia and epidural anesthesia, as well as lumbar punctures, are performed in this region here. And the reason for that should be obvious. The anatomy allows a needle to be inserted through the vertebrae and into the epidural space, or even into the cerebrospinal fluid that's surrounding the cauda equina. And because the cauda equina is so small compared to the spinal cord, there's much more room here, and you don't risk collateral damage to the spinal cord itself. Nerves from the cervical region here innervate the chest, head, neck, shoulders, arms, hands, and the diaphragm. Nerves from the thoracic region, which is here, extend to the intercostal muscles of the rib cage, the abdominal muscles, and the back muscles. The lumbar spinal nerves that come out in this region innervate the lower abdominal wall and parts of the thighs and legs. Nerves from the sacral region here extend to the thighs, buttocks, muscles, skin of the legs and feet, and the anal and genital regions. There's 31 pairs of spinal nerves that branch out from the spinal cord, and these are the main divisions: the cervical, thoracic, the lumbar, and sacral. It's important to note that, again, the spinal cord itself ends at about L1, the first lumbar vertebrae, but the spinal nerves continue to extend down lower in the spinal canal, and where they leave the vertebral column at a different location. The spinal cord sits inside stacked vertebrae. A cross-section shows the two types of nervous tissue, which is white matter here and gray matter here. Gray matter appears gray because it's lack of myelin that we talked about. It contains mostly the cell bodies of motor neurons and interneurons. The H-shaped mass that you see, that's shaped kind of like an upside-down butterfly, is divided into two sets of horns. The posterior or dorsal horns, shown here, and the ventral or anterior horns, shown here. White matter appears white, obviously, because of the abundance of myelin. It contains bundles of axons called tracts that carry impulses from one part of the nervous system to another. Let me go back a second here. The central canal, which is shown here, this little canal in the center of the spinal cord, carries cerebral spinal fluid. The epidural space, which is the fatty area shown in yellow here, all the way around, this is the epidural space, lies between the outer covering of the spinal cord and the vertebrae. So here's the outer layer of the spinal cord called the dura mater, and this is the bony vertebrae. The space in between is called the epidural space, and it's full of fat as well as blood vessels and connective tissue. spinal nerves that are shown here, this is a spinal nerve, and these are the nerve roots. So the spinal nerves travel through gaps between the vertebrae and attach to the spinal cord by way of the dorsal and ventral roots. So of course this would be the dorsal root, and this is the ventral root. The dorsal or posterior nerve root contains fibers that carry sensory information into the spinal cord. It enters the dorsal horn here of the spinal cord. Cell bodies of the dorsal neurons are clustered in a knot-like structure called the ganglion, which is here. And that again is these are called the dorsal root ganglion. Fibers in the ventral or anterior nerve roots exit from the ventral horn here. They exit out here to carry motor information out of the spinal cord. A spinal nerve which is here, this is a spinal nerve, is a single nerve resulting from the fusion of the dorsal and ventral nerve roots. It's a mixed nerve. It contains both sensory and motor fibers, and that means it can conduct impulses in two directions, shown here. This direction coming into the spinal cord is called afferent, and impulses that are leaving the spinal cord are called efferent. The spinal cord is protected by the bony vertebrae and also by 3 layers of fibrous connective tissue called the meninges. The pia mater, shown here, if I can find my pointer, there we go. The pia mater is the innermost layer. It's a transparent membrane that clings to the surface of the brain and spinal cord. It contains blood vessels. The arachnoid mater lies between the dura mater and the pia mater, and it's a delicate layer that resembles a cobweb. The dura mater is the tough, dense outermost layer. The subarachnoid space lies between the arachnoid mater and the pia mater, and it's filled with cerebral spinal fluid. So this area in between the pia and the arachnoid is called the subarachnoid space. It's full of blood vessels here and also cerebral spinal fluid. Bundles of axons called tracts lie within the white matter, not the gray matter. They serve as routes of communication to and from the brain. All nerve fibers in a single tract have a similar origination, destination, and function. So you have ascending and descending tracts. Ascending tracts convey sensory signals such as pain up the spinal cord to the brain. Ascending tracts are the dorsal column, which are shown here. So these are ascending. The spinal cerebellar tracts, which are here, and the spinal thalamic, which are here. It's interesting that these are all on the right side of the spinal cord. The dorsal column relays sensations of deep pressure, vibration, and those needed to create awareness of the body's position. It's called proprioception. The spinal cerebellar tract is responsible for proprioception also, and the spinal thalamic tracts down in this region here relays the sensations of temperature, pressure, pain, and touch. Descending tracts over on the left side of the spinal cord conduct motor impulses down to the skeletal muscles. These include the corticospinal tracts and the extrapyramidal tracts. So sometimes the corticospinal tracts are called the pyramidal tracts, and it's the ones here in red or maroon, and all of the others are in the yellow or orange tan colored, are called the extrapyramidal. The corticospinal tracts are responsible, so the red ones are responsible for fine movements of the hands, fingers, feet, and toes on the opposite side of the body. And the extrapyramidal tracts are groups of tracts associated with balance and muscle tone. Neuroanatomy is interesting because it allows physicians to predict what a person might experience if some part of the spinal cord is affected with a tumor, a stroke, or some traumatic event. This slide is an illustration of a nerve. It consists of fascicles and blood vessels that you can see. Here is a blood vessel, and here is a fascicle. All of these are surrounded by a dense connective tissue called the epineurium here, and so you can see it's this layer of dense connective tissue. Nerve fibers or axons exist in bundles called fascicles. Several fascicles are grouped together and wrapped in a connective tissue sheath called the perineurium here. Within the fascicle, each axon or nerve fiber is wrapped in a loose connective tissue called endoneurium. The number of fascicles contained in a single nerve varies from just a few to a million. Most nerves contain both sensory and motor fibers and are called mixed nerves, and these nerves can transmit signals again in both directions. A few nerves, such as the olfactory and optic cranial nerves, are sensory nerves only, and they contain only afferent fibers. They carry sensations toward the spinal cord only. Others are dedicated motor nerves and contain only motor or efferent fibers carrying messages to muscles and glands. Figure B down below here is a micrograph of an actual nerve in cross-section and shows the structures that we talked about. So this is a diagram that shows the different areas that we talked about earlier of the spinal cord and the spinal nerves that radiate out from the spinal cord. The first cervical nerve exits the spinal cord between the skull and the axis up here. The other nerves pass through the holes in the vertebrae called the intervertebral foramina. Outside the spinal column, each spinal nerve forms branches. Some of these branches form nerve networks called plexi, or singular is plexus. The four major plexi are the cervical plexus here, the brachial plexus, the lumbar plexus, and the sacral plexus. The cervical plexus contains nerves that supply the muscles and skin of the neck, tops of the shoulders, and part of the head. The phrenic nerve, which stimulates the diaphragm for breathing, is also located in the cervical plexus. The brachial plexus innervates the lower part of the shoulder and arm. Key nerves in this region include the axillary, radial, ulnar, and median nerves that run all the way down to the hand and tips of the fingers. The lumbar plexus supplies the thigh and leg. A key nerve in this region is the large femoral nerve. The sacral plexus is formed from fibers from nerves L4, L5, S1, and S1 through S4. The sciatic nerve which is the largest nerve in the body, arises here and runs down the back of the thigh. This is a common area for disc herniations down in this region here, which is why one of the common symptoms of a herniated disc is radiating pain down one or both legs, and that is the sciatic nerve, and it's sometimes called sciatica. Again, there's 31 pairs of spinal nerves, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. And some of these group together to form the plexi. Reflexes are a quick, involuntary, predictable response to a stimulus. There's a good overview video of somatic reflexes here that you should watch. You can access that again through the lecture slides. Reflexes, they use a neural circuit called a reflex arc. These bypass the regions of the brain where conscious decisions are made, which is why someone only becomes aware of a reflex after it has occurred. Autonomic, so there's somatic reflexes and autonomic reflexes. Autonomic reflexes involve secretion from glands or the contraction of smooth muscle, such as dilation of the pupil in the eye. These reflexes are governed by autonomic neurons, and we'll get to that in a minute. Somatic reflexes involve the contraction of a skeletal muscle after being stimulated by a somatic motor neuron. So even though the somatic nervous system is typically called the voluntary nervous system, somatic reflexes never reach any level higher than the spinal cord where the spinal nerve enters and exits, which is why it's called a reflex. Somatic reflexes protect the body against harm and maintain posture. So they involve these steps. You have in step 1, here you have somatic receptors located in the skin, a muscle, or a tendon. In this case, it's stretch receptors in the tendon that detect a sensation, such as stretching of the tendon when the patellar tendon is tapped. Step 2, you have an afferent sensory nerve that sends a signal directly to the spinal cord. So that's shown here. The impulse immediately passes to a motor neuron because it's coming from a specific type of receptor. It does not ascend in any of the tracks that we talked about. Instead, an interneuron connects it directly to a motor neuron, and the motor neuron then leaves the spinal cord through the ventral root and goes back to the muscle, causing it to contract, and that produces a slight kick that we all see with the patellar tendon reflex. This explains why doctors always check reflexes in clinic, is because if there is some pathology of the spinal cord that's affecting this region here, reflexes will be non-existent or hyper. The brain, shown here, is divided into 4 major regions: the cerebrum, which is all of this tan area, the diencephalon, that's labeled here, the cerebellum, which is down here in this region. Here's the diencephalon, and the brainstem, which is basically all of these structures together. There's 3 parts to the brainstem. You have the midbrain, the pons, and the medulla. And this should say, instead of midbrain, it should say brainstem here. That's a typo. The cerebrum is the largest portion of the brain. Obviously, you can see that it extends all the way around here. Thick ridges called gyri or gyri are these ridges in between the shallow grooves called sulci. Deep sulci are called fissures. So if they're really deep, you can see here, this would be a fissure. The more shallow ones are called sulci, like this. The diencephalon sits between the cerebrum and the midbrain. It's a deeper structure that's only visible if you section the brain, either a frontal or sagittal section. The cerebellum is the second largest region of the brain, and it contains more neurons than the rest of the brain combined. The brainstem is the final part. It consists of the midbrain here, the pons, and the medulla oblongata. And the cranial nerves all extend from the brainstem except for the first two. There's a deep groove called the longitudinal fissure that divides the cerebrum into right and left cerebral hemispheres. You can't see that here on this view. A thick bundle of nerves called the corpus callosum runs along the bottom of this fissure. The brain contains gray and white matter, just like the spinal cord. The gray matter is the most superficial, and it covers the cerebrum and cerebellum in a layer called the cortex, and its function is thought, learning, and reasoning. Surface folds allow more gray matter to be packed into the skull, so that's the reason that you have sulci and gyri, is to increase surface area for more gray matter. The brain contains 4 chambers called ventricles that are shown here. You have the lateral ventricles here. There's 2 of those. The third ventricle and the fourth ventricle. So here's the third ventricle in green, fourth ventricle here. The lateral ventricles are in blue. The 2 lateral ventricles arch through the cerebral hemispheres. There's one on the right and one in the left hemisphere. Each of the lateral ventricles connects to the third ventricle through a passageway that's right here. A canal called the aqueduct of Sylvius is here. Leads from the third ventricle to the fourth ventricle. It narrows after the fourth ventricle to form the central spinal canal here. And that extends all the way down through the spinal cord. The bones of the skull help protect the brain from trauma. So inside of the skull, just like in the spinal cord that we talked about, there's 3 layers. Again, it's the same thing. You have the dura mater, which is here, and this layer is split in the brain right in the middle. And so here's the outermost and then an inner layer, and we'll talk about why that is significant in a minute. The arachnoid mater is this blue colored layer here, and then the very fine pia mater, which is down here right on the surface of the brain. So again, the subarachnoid space contains blood vessels as well as cerebrospinal fluid. The subarachnoid space is in here, right on top of the pia mater. So the dura mater, again, consists of 2 layers. The periosteal layer is this one, the outermost, and that's attached to the inner part of the skull. And then you have the inner meningeal layer, which is here. The inner meningeal layer forms the outer covering of the brain, and it continues as the dura mater of the spinal cord. The duramater separates to create spaces in the brain called dural sinuses, and these are full of venous blood. So this is a dural sinus here. These sinuses collect blood that is passed through the brain and is on its way back to the heart. A subdural space separates the dura from the arachnoid. The subarachnoid space separates the arachnoid from the pia mater. In some places, the dura mater extends inward and separates major portions of the brain. An example of that is the falx cerebri here. So you have the meningeal layer of the dura mater that dives in and fuses together to form this thick falx cerebri, which dips into the longitudinal fissure, and it separates the right and left hemispheres of the brain. Elsewhere, you have the tentorium cerebella that extends over the top of the cerebrum, separating it extends over the top of the cerebellum, separating it from the cerebrum. And I don't have a picture of that. This slide shows the circulation of cerebrospinal fluid, which is obviously very important. It's a clear, colorless fluid that fills the ventricles and central canal, and it also bathes the outside of the brain and the spinal cord. The choroidal plexus in each lateral ventricle secretes CSF, cerebrospinal fluid, which constantly flows through the central nervous system. Cerebrospinal fluid provides nourishment, glucose, and protein. It removes metabolic waste. It cushions the brain against minor trauma. It provides immune protection, and it also carries oxygen and other waste gases like carbon dioxide. The brain monitors the level of CO2 in this CSF and triggers responses to help maintain body homeostasis. So CSF production goes as follows. The choroidal plexus within each lateral ventricle secretes cerebrospinal fluid. It should be noted that the choroidal plexus is also present in the third and fourth ventricles that you can see here. There's a small amount in the fourth ventricle. It's also in the third ventricle. The CSF then flows. So if we begin in the lateral ventricles, it's formed here, and then it flows down into the third ventricle through this foramen called the foramen of Magendie. The choroidal plexus in the third ventricle adds more fluid, and then it flows down through the aqueduct of Sylvius into the fourth ventricle, where still more CSF is added from the choroidal plexus there. Some of the CSF flows through two tiny openings called foramina. Again, you can see those here and also here. In step 5, you can see if it flows through here, it goes around the cerebellum. It can come up and also go to encircle the brain, and it can also go down into the spinal cord to surround the spinal cord. So it has the potential to go all the way around the brain like this and down around the pituitary gland here. It surrounds the hypothalamus, which is this region, and it comes down in front of the brainstem as well. It can go down right in the central canal, so it bathes the entire central nervous system. Now, how does CSF that's full of waste products get back into the venous blood so it can return to the general circulation to be filtered in the kidneys and liver and whatnot? And the answer is it's reabsorbed into the venous bloodstream by projections of the arachnoid mater in the dural sinuses called arachnoid villi. These are special projections that only occur in the dural sinus superiorly, and this is an example. And the CSF passes up, and there's special cells that are one-way cells that only allow CSF to pass from the inside out into the venous blood in the dural sinus. And the previous slide showed a good example of these as well. You have CSF in this layer, in the subarachnoid space, and in some areas you have the arachnoid villi that puncture or penetrate the dura mater, the meningeal layer of the dura, and come out here and protrude into the dural sinus. And the CSF can then pass up through these arachnoid granulations or arachnoid villi and pass into the venous blood. The brainstem consists of the midbrain here, the pons, and the medulla oblongata. The midbrain contains tracts that relay sensory and motor impulses. It also contains centers for the auditory and visual reflexes, as well as clusters of neurons that are important for muscle control. So that is the midbrain. The third and fourth cranial nerves are projecting from the midbrain. The pons, shown in green here, contains tracts that convey signals to and from different parts of the brain. And again, several important cranial nerves arise in this area. You can see the large fifth cranial nerve, as well as the sixth and seventh cranial nerves down here. The medulla oblongata attaches the brain to the spinal cord, and it relays sensory and motor signals between the brain and the spinal cord. And it's shown here in this area. It contains the cardiac center, which regulates heart rate, the vasomotor center, which controls blood vessel diameter and, in turn, blood pressure, and two respiratory centers that regulate breathing. The medulla also houses reflex centers for coughing, sneezing, swallowing, and vomiting. And several cranial nerves either begin or end in the medulla. Essentially, cranial nerves 8 through 12 arise from the medulla oblongata. This slide shows three important areas. You have the cerebellum shown here, the thalamus, and the hypothalamus, which together make up the diencephalon. The cerebellum houses more neurons than the rest of the brain combined. It's connected to the cerebral cortex by approximately 40 million neurons, and it receives and processes messages from all over the brain. You can think of it as sort of a fine-tuning mechanism for all of the functions of the cerebrum. It monitors body movements and sends messages for balance, coordination, and posture. It stores the information necessary for muscle groups to work together to perform smooth, efficient, and coordinated movements. It evaluates sensory inputs such as touch, spatial perception, and sound. People with cerebellar dysfunction have a spastic gait, poor balance, jerky movements, and tremors. They also have poor impulse control and overreact emotionally. A region deep inside the brain, the diencephalon, which is shown here, consists of several structures, with the chief ones being the thalamus and hypothalamus. The thalamus is shaped like two eggs. It resides on the top of the brainstem, and it acts as a gateway for nearly every sensory impulse traveling to the cerebral cortex. Most of you don't know about switchboards, but in the early days of telephone communication, there was a switchboard person, and the thalamus is like a switchboard. every nerve that comes into or leaves the entire cerebral cortex passes through the thalamus. It processes and filters impulses. It transmits some, but not all, up into the cerebral cortex. It also relays messages regarding certain complex movements, and it's involved also in memory and emotion. The hypothalamus is tiny in size, but it influences nearly every organ of the body. It's very small, only about the size of a pea. It controls the autonomic nervous system. It contains centers for hunger, thirst, and temperature regulation. It controls the pituitary gland, here sometimes called the master gland. It's involved in multiple emotional responses that include fear, anger, pleasure, and aggression. A set of interconnected nuclei in the brainstem called the reticular formation is very important. Fibers extend from the RAS, called the reticular activating system, which is shown in blue here. So fibers extend from the RAS up into the cerebrum after passing through the thalamus, which is shown here. Fibers also extend to the cerebellum, and they can also go back down into the spinal cord. One component of the RAS receives sensory input from the eyes and ears. So you have visual as well as auditory input that comes in. The visual input comes in obviously through the optic tracts and enters the RAS, and then you also have auditory input coming in through the 8th cranial nerve, which is in the pons midbrain area right down in here, and that also enters the RAS. This important structure receives signals from visual and auditory input, and it filters out insignificant signals such as routine noises, background noise, and it sends impulses to the cerebral cortex so the mind remains conscious and alert. Drugs called hypnotics depress the RAS and they induce sleep, and that's what general anesthesia does. So general anesthesia is a hypnotic that suppresses the RAS. It also has tracts that extend to the spinal cord that are involved in posture and equilibrium. Basically, consciousness begins in the RAS. Other components of this important structure include cardiac and vasomotor centers of the medulla, which are responsible for heart rate and blood pressure. The cerebrum is the largest portion of the brain. It's divided into 5 major lobes, each of which is named for bones of the skull that lie directly over them. So you have the frontal lobe here, covered by the frontal bone. You have, the frontal lobe governs voluntary movements, memory, emotion, social judgment, decision-making, reasoning, and aggression. And it's also the site for certain aspects of our personality. The parietal lobe is shown here. Actually, it's more the green section. Frontal lobe is here. The parietal lobe is concerned with receiving and interpreting bodily sensations, such as touch, temperature, pressure, and pain. It also governs proprioception, which is the awareness of our own body and body parts in space and in relation to each other. The occipital lobe here is concerned with analyzing and interpreting visual information. And the temporal lobe, shown here, governs hearing, smell, learning, memory, emotional behavior, and visual recognition. The insula, which is hidden and it's only exposed by separating along the longitudinal fissure here. This is called the insula. It plays a role in many different functions, including perception, motor control, self-awareness, and cognitive functioning. So these are the 5 major lobes of the brain. The frontal lobe here, the parietal lobe, the occipital lobe, the temporal lobe, and the insula. The bulk of the cerebrum is shown in this slide as white matter, which consists of bundles of myelinated nerve fibers called tracts that we talked about. So all of these tracts in the spinal cord come up into the brain to contribute to the white matter, which is shown all in here. These tracts carry impulses from one part of the cerebrum to the other, or from the cerebrum to other parts of the brain or spinal cord. Most tracts passing from one hemisphere to the other travel through the corpus callosum, shown in green here. This allows the brain's 2 hemispheres to communicate with each other. The surface of the cerebrum, called the cerebral cortex, is out here on the surface, obviously, and is gray matter. Gray matter makes up only about 40% of the brain's mass. Masses of gray matter called basal nuclei or basal ganglia lie deep in the cerebrum and also in the spinal cord, well, the brainstem and the spinal cord, and play a role in the control of movement. The basal ganglia is this area here, right around in the thalamus region, and this is what's involved in diseases such as Huntington's disease and Parkinson's disease. Other tracts carry information back and forth between the brain and spinal cord, and that's shown here. These tracts are extensions of the ascending sensory spinal thalamic and tracts and the descending motor or corticospinal tracts. The tracts in the brainstem, they cross in the brainstem here, with the right side of the brain sending impulses to the left side of the body and vice versa. And this is why if a person has a stroke, it affects either the right or left hemisphere, but then symptoms also involve the opposite side of the body. Sensory fibers transmit signals up through the spinal cord to the thalamus, which then forwards them, so this is sensory again, to the post-central gyrus. So here's the central gyrus, or fissure, and you have the primary somatic sensory area, or the post-central gyrus here. This is responsible for somatic sensation in the body. It receives impulses of heat, cold, and touch from receptors all over the body. Because of decussation, or crossing over, the right postcentral gyrus receives signals from the left side of the body, and the left postcentral gyrus receives signals from the right side of the body. The somatic sensory association area, shown here in the parietal lobe, allows us to pinpoint the location of pain, identify a texture, and to be aware of how our limbs are positioned relative to each other. The primary somatic motor area is the precentral gyrus. So again, you have the central sulcus or fissure here, and this is the precentral gyrus here. This is the primary motor area. Neurons in the motor association area, which is here in the frontal lobe, determine which movements are required to perform a specific task and send the appropriate signals to the precentral gyrus. Neurons in the precentral gyrus then send impulses through the motor tracts in the brainstem and spinal cord to the skeletal muscles where movement occurs. So again, the primary somatic motor area is the precentral gyrus, and the motor association area is where willful voluntary movement originates, and it then goes to the primary motor, the primary motor precentral gyrus that then sends impulses through the motor tracts to the brainstem and spinal cord to the different muscles for movement. This diagram illustrates how the different areas of the brain synchronize to produce a willful action. To start with, you have written words. Let's say somebody's reading, and written words stimulate the primary visual cortex here. So this is step 1. Here, the primary visual cortex. In step 2, the angular gyrus, which is here, which is part of the parietal lobe, translates the written words into a form that can be spoken. This is why injuries to the parietal lobe here in this area produce aphasias, which are both receptive and expressive. In step 3, the impulse goes to Wernicke's area, which is here, where words are formulated into phrases that comply with learned grammatical rules. In step 4, we go over here to Broca's area, which plans the muscle movements required for the larynx, tongue, cheeks, and lips to form the words. It then sends the appropriate impulses to the primary motor cortex here. The primary motor cortex sends impulses to the muscles necessary to pronounce a word. So this is the process that occurs when a person is reading aloud. Cerebral processing is associated with the 5 major physical senses. You have, number 1, the primary gustatory complex, which handles the interpretation and sensation of taste. And so that would be right here. This is the primary gustatory area. Number 2 is the primary visual cortex. So this is vision. It's responsible for sight. It governs the recognition of size, color, light, motion, and dimension. The visual association area, which extends out here in the orange, interprets the information acquired through the primary visual cortex and allows us to recognize familiar objects. Area 3 is the primary auditory area, which is here, and then the auditory association area, which is out in this region. This is on the temporal lobe. It's responsible for hearing, and the association area gives us the ability to recognize familiar sounds, including a person's voice. And this is why people who are blind, if it's not a cortical blindness, the brain that's normally dedicated for vision, the brain is a very plastic organ, and the nervous resources that are normally dedicated to vision actually move over into the auditory region and other regions and magnify those senses. This is why a person who is blind, sometimes they can hear much better, or they can feel much better than the average person. I grew up with a brother who's totally blind, and it's true. He can hear things and recognize sounds and associate them with things very, very perceptively that I could never do. We also have the olfactory association area, which interprets the sense of smell, and that is, I don't know if that's shown here, but that is essentially this area down here. And then finally we have the primary somatic sensory area, which is here. This again is the post-central gyrus, and it interprets and processes the sense of touch. So these are all of the 5 senses and where they are located in the brain. Then of course we have other areas that we talked about. Sensory speech area is Wernicke's area here. We have the motor speech area, which is Broca's area here. And the primary motor cortex, or the precentral gyrus, is here. It's important to be able to know where these different areas are and what their basic functions are. And this brings us to a homunculus, which is shown here. And these funny people, a homunculus actually comes from the Middle Age, from the Renaissance period, and these are drawings of disproportionate figures, and it illustrates different functions. The relative size of each structure in the homunculus correlates with the degree of innervation. Structures or processes requiring more nervous system resources are larger than others. Note that the hands, face, and pharyngeal structures, such as the larynx for voice and song, demand much more neural resources, indicating that these are the most complex in the body. So the motor homunculus, red, which is the one here on the left, illustrates cerebral mapping for motor control of different body structures. And you can see all of those here that correspond with this funny little man called the homunculus. The sensory homunculus here in blue shows the cerebral mapping for various sensory stimuli, and it's similar to the motor one, but there are some differences. So, to reiterate, again, the homunculus showed there was the motor and sensory. Now, those did not indicate the right and left hemispheres of the brain. They simply were showing the motor and sensory areas, which would be, here's the central sulcus here on each side of the brain. And so the primary, the precentral sulcus is here, and the postcentral gyrus is here. Again, these are not right or left, but they're on both sides of the brain. However, the hemispheres of the brain, you also have a division that goes right down here, and the hemispheres are dedicated to different things. The left hemisphere over here on this side is the more analytical side. It focuses on language, and the types of reasoning used in math and science. The right hemisphere is more concerned with creativity and spatial ability, such as art. The two hemispheres communicate through the corpus callosum that allows the smooth integration of information. We're now at the peripheral nervous system. Because the nervous system performs so many functions, it's helpful to further subdivide the peripheral nervous system as shown here. So we just talked about the central nervous system, and it was pretty straightforward, but the peripheral nervous system has many more divisions, so we're going to talk about those now. We have 12 pairs of cranial nerves that arise directly from the brain to relay messages to the rest of the body. Each cranial nerve is identified by a name and a Roman numeral beginning in the anterior portion of the brain. So cranial nerve number 1 is the olfactory nerve, which begins right here, right from the brain. Cranial nerve number 2 is here. It crosses over right after the pituitary, or posterior to the pituitary gland, and it's shown here. So the first and second cranial nerves actually arrive directly from the brain itself. Some cranial nerves contain only sensory fibers, some contain primarily motor fibers, and others contain both. And so the 12 cranial nerves are the olfactory, number 1, number 2 is the optic, number 3 the oculomotor, number 4 is the trochlear nerve, number 5 is the trigeminal nerve, here. Number 6 is the abducens nerve. Number 7, so here's number 6. Number 7 is the facial nerve. Number 8 is the vestibulocochlear nerve that goes to the inner ear. Number 9 is the glossopharyngeal nerve. Number 10 is the vagus nerve that goes down to the smooth muscle and organs in the thorax and abdomen. Number 11 is the spinal accessory nerve that goes to the muscles of the neck. And finally, number 12 is the hypoglossal nerve. It's interesting to note that of the cranial nerves, cranial nerve number 2, 3, 4, and 6 are dedicated only to the eye. And then branches from the 5th and 7th cranial nerves, many of them also go to the eye. So the eye is a very complex structure that demands a large part of the cranial nerves. There's an excellent video overview of the cranial nerves here that you should all take a few minutes to watch. Visceral reflexes are similar to somatic reflexes that we looked at earlier, in that they occur before we are consciously aware, and they also occur without any voluntary or willful input from our brain. Visceral reflex activities include the secretion of digestive enzymes, constriction and dilation of blood vessels for maintenance of blood pressure, and the secretion of hormones, just to name a few. These activities occur without awareness or control, so they're autonomous. It is also called the visceral motor system because it targets the organs or viscera of our body. The autonomic nervous system asserts control through visceral reflexes similar to the somatic reflexes, but these affect an organ or a gland instead of a skeletal muscle. All visceral reflexes follow similar steps, and this illustration shows the visceral reflex arc responsible for the regulation of blood pressure on the left, and the pupillary light reflex on the right. There's also a good animation here. It's short, but it's very good. It's an overview of visceral reflexes, and you should watch that. So receptors, in this example, it's right in the carotid artery near the carotid bifurcation, where the common carotid splits into the internal and external carotid. The receptors in the eye are in the retina, shown here. So in step 1, receptors detect a change in body conditions. Carotid baroreceptors detect a rise in blood pressure, and the retinal rods and cones sense increasing brightness in the left eye in this example. In step 2, afferent neurons, remember I said afferent, transmit information about this change to the central nervous system. So that's what this is. You have it going here into the brainstem, and this one travels also along the third cranial nerve to the midbrain. The glossopharyngeal nerve up here transmits the information to the medulla oblongata. So in this example, we're midbrain, this one, we're medulla. This is the glossopharyngeal nerve. This is the oculomotor nerve. The brainstem processes the information and transmits a signal along an efferent nerve. In the blood pressure example on the left, it's the vagus nerve that is the motor nerve. The vagus nerve sends a signal to the heart's pacemaker, the SA node, to slow its rate. The oculomotor nerve transmits its signal to the intraocular muscles of the iris and ciliary body, called the intraocular muscles. So it comes back along the third cranial nerve, the oculomotor nerve. I think earlier I said that the afferent nerve was the third. It's actually the optic nerve, the second. And it goes in to the midbrain. And third cranial nerve, the oculomotor nerve, carries the signal back out through the ciliary ganglion out to the intraocular muscles and stimulates them to contract. The effector organ receives the message and responds. So in this example, it's the SA node in the atria of the heart, and in this example, it's the intraocular muscles of the eye. So, the somatic pathway of the peripheral nervous system, the neuron cell bodies lie within the spinal cord. So that's shown here in the gray matter. And note that it's the ventral horn of the spinal cord. A single myelinated axon extends from the brainstem or spinal cord to a skeletal muscle. It's very straightforward and simple. Note that the somatic nerves are very heavily myelinated because speed is important to stimulate muscle contraction. At the target muscle, the neurotransmitter acetylcholine, shown here, is released to cause muscle contraction. In the autonomic pathway of the peripheral nervous system, it's a bit more complicated. A myelinated preganglionic neuron extends from the brainstem or spinal cord out to a ganglion. Again, it begins in the ventral gray matter of the spinal cord. It comes out, passes through. Again, this is myelinated. In the ganglion, out, it can be near the spinal cord or out near the target organ, depending on if it's a sympathetic or parasympathetic ganglion. In the ganglion, it synapses with a postganglionic neuron, which is here, this guy. and the neurotransmitter acetylcholine is released, and that's how the signal goes from preganglionic synaptic knob to postganglionic dendrite here. The axon of the unmyelinated, and note that this is unmyelinated, postganglionic neuron extends to the target organ, and it's unmyelinated because speed is not as important. The neurotransmitter here releases different neurotransmitters. If it's a parasympathetic neuron, it will release acetylcholine. If it's a sympathetic fiber, it will release norepinephrine, or very rarely, sometimes the sympathetic system will also use acetylcholine. So there's a good video overview of the sympathetic system that you should look at. The sympathetic preganglionic neurons begin within the spinal cord. From the cell bodies, myelinated fibers reach the sympathetic ganglia that are shown here. Here's the cell bodies in the ventral aspect of the gray matter of the spinal cord. From the sympathetic ganglia exist in chains along both sides of the spinal cord, and they tend to be quite close to the spinal cord or the cell bodies of the sympathetic neurons. The preganglionic neurons are short. That's these here. They're relatively short, but not all preganglionic neurons synapse in the first ganglion they encounter. So some of them come in to a ganglion, and then they might dive up to another one. And these synapse with other ganglia at different levels, and these are called splanchnic nerves. Unmyelinated, again, remember these are unmyelinated, postganglionic fibers leave the ganglia and extend to the target organs. Postganglionic fibers tend to be long and branched, and these branches synapse with multiple postganglionic receptors in different target organs, and this allows one postganglionic neuron to excite multiple end organs simultaneously. So you can see this one goes to the liver, the stomach, the spleen, and the pancreas, all from one postganglionic neuron of the sympathetic system. This allows it to service many organs at once, which is why the sympathetic system or the sympathetic division can ignite such an immediate, widespread reaction, and we sometimes call this the fight or flight response. You can see that it innervates virtually every major organ of the body. This illustration shows how splanchnic nerves interconnect the sympathetic ganglia to magnify the output. So in this example, let me find my little pointer here. Again, this shows how simple the somatic system is. It goes from the ventral gray matter right out here to the skeletal muscle, no big deal. But the sympathetic and parasympathetic branches of the autonomic system are much different. This is the sympathetic example, and you have here, it begins and it comes here, and one branch goes, stops here in the ganglia to connect with another nerve that then goes out, but then another branch goes up to a ganglion more superior, another one goes out laterally, and another one dives down here. So these nerves that go to different ganglia are called splanchnic nerves, and this interconnection allows for this magnified response of the sympathetic system. The sympathetic system also utilizes the adrenal glands, which are part of the endocrine system. Sympathetic preganglionic fibers pass through the outer cortex, which is here. This is the outer cortex area of the adrenal gland, and terminate in the center, called the adrenal medulla. So the center area here is the adrenal medulla. When stimulated, the adrenal gland secretes a mixture of epinephrine, also known as adrenaline, and norepinephrine directly into the bloodstream. So again, you have the cells in the adrenal medulla that secrete epinephrine and norepinephrine right into the bloodstream, and this further magnifies the sympathetic response, and it's critical for survival. Fight or flight is the system that kicks in for our survival, so this is why it's important for it to be so magnified. Now, it's important to remember that this is just the medulla of the adrenal gland, so it's part of the sympathetic nervous system. It's the cortex of the adrenal gland that's part of the endocrine system, and the cortex is very, very interesting. It secretes some important hormones that we'll discuss in the endocrine system. Parasympathetic reflexes are shown here. There's a good video overview of this as well that you should look at. Neurons of the parasympathetic division arise from the brain, the cervical, brainstem, actually the brainstem area, not the cervical, and the sacral area of the spinal cord. This is why the division of the parasympathetic autonomic system is also called the craniosacral division. Parasympathetic fibers leave the brainstem by joining either the oculomotor, cranial nerve 3, facial, cranial nerve 7, glossopharyngeal 9, or vagus nerve number 10. That's all up here in the brainstem. The vagus nerve carries about 90% of all parasympathetic preganglionic fibers. The ganglia of the parasympathetic division reside in or near the target organ, as opposed to the sympathetic ganglia. The parasympathetic preganglionic fibers are long, and the postganglionic fibers are short. The preganglionic fibers, most of them are not myelinated, or very, very sparsely myelinated, and the postganglionic fibers are not myelinated. Because the ganglia are more widely dispersed, the parasympathetic division produces a more localized response than that of the sympathetic division. However, the parasympathetic responses can still have a dramatic systemic effect especially if the vagal nerve is involved. And some of you may have experienced like a vasovagal reaction if you see something that really is gross, or that you feel sick to your stomach and all of a sudden you feel lightheaded and feel like you're going to pass out. That is a massive vagus response of the parasympathetic system. You can see how the vagus nerve here reaches out to innervate all of the major organs that the sympathetic system does. Now, it's again important to remember that the ganglia, which are mainly up in the cranial area, are located very near the organ. They're not in close to the spinal cord like the sympathetic system. Okay, so this is a table showing, comparing and contrasting the two branches of the autonomic nervous system. Both the sympathetic and parasympathetic, they work at the same time, and they provide a background level of activity called autonomic tone. It's the balance between sympathetic and parasympathetic activity that constantly changes depending on the body's needs. But it's important to remember that it's not one or the other. They're both going at the same time, and it's the relative strength of one to the other that determines the autonomic tone. And these are the major differences of the sympathetic and parasympathetic, so you can review these. So this is a diagram. I like it because it puts everything into one diagram for comparison. The two divisions of the autonomic nervous system, again, they exert opposite effects. There are some exceptions. The sympathetic division, for instance, dilates blood vessels, leading to skeletal muscles, but it constricts blood vessels, leading to most organs. So, but the overall effect is the same. The sympathetic is fight or flight. Parasympathetic is rest and repair, or has a more calming effect. Two factors determine the effect of each division. So here we have the sympathetic division, and this is the parasympathetic division. This example here, the second example, is much less common than the first. The first one here is what we normally see. The only time that the sympathetic system uses cholinergic receptors that I believe is in some glands and also in blood vessels, and this shows that. So two factors determine the effect of each division. It's the type of neurotransmitter released, whether it's acetylcholine or norepinephrine, and it's the type of receptor on the target cells, whether they're adrenergic or cholinergic. So it's important to remember that all of the preganglionic neurotransmitter is always acetylcholine, and that's shown here. So these are called the cholinergic receptors, and if the receptors are part of the parasympathetic, we sometimes call those nicotinic receptors. So the two major neurotransmitters are acetylcholine, which is present in all of the first, the preganglionic junction, and the second junction, it can be norepinephrine, is shown here. It can be acetylcholine, shown here, or it's always acetylcholine in the parasympathetic system. So fibers that secrete acetylcholine are called cholinergic fibers, and fibers that secrete norepinephrine are called adrenergic fibers, is shown here. Adrenergic, cholinergic, and cholinergic. The effects of the sympathetic division last longer than those of the parasympathetic division because acetylcholine is disposed of more quickly after it's released than is norepinephrine. Neurotransmitters bind to receptors on the effector cells of the target organs. Acetylcholine binds to cholinergic receptors, and that's shown here. And norepinephrine binds to adrenergic receptors here. And in very few cases, again, you can have cholinergic receptors that respond to the sympathetic system. Cholinergic receptors may be one of two types, and the different types of receptors determine the effect produced by a neurotransmitter. The variable responses produced by muscarinic receptors allows acetylcholine to stimulate intestinal smooth muscle, whereas it inhibits cardiac muscle. So you have nicotinic receptors, which are all somatic receptors and all autonomic preganglionic. These are called nicotinic. Then you have muscarinic receptors, which are all postganglionic parasympathetic here, and these are all parasympathetic postganglionic receptors. While the parasympathetic system has only cholinergic postganglionic fibers and muscarinic receptors, the sympathetic system has both adrenergic and cholinergic, as I've mentioned several times. Then there's a further subdivision of the adrenergic receptors that includes both alpha and beta subtypes. So these can be alpha or beta. The binding of norepinephrine to alpha receptors in blood vessels causes them to constrict. The binding of norepinephrine to beta receptors in the heart and skeletal muscles causes the blood vessels to dilate. Even though it's the same neurotransmitter, it's a different effect because of the receptor. Both alpha and beta receptors contain subtypes that can cause exceptions to these principles. For example, beta receptors in the heart, beta 1, stimulate cardiac muscle contraction, while beta receptors in the airways, called beta 2 receptors, cause relaxation of smooth muscles. So this is why doctors prescribe beta blockers to lower blood pressure, and they prescribe beta agonists, drugs that work on the beta 2 receptors, specifically to treat asthma, because it dilates the smooth muscle in the airway. There's a very good video overview of the autonomic nervous system here that you should watch, and you can access that through the lecture slides. So this wraps things up for this lecture. It's complicated, but hopefully I laid it out in a way that will make it easy to understand. And I look forward to discussing this more with you in class. Thanks, and have a great day.